You may have seen the headlines. Scientists can now take an ordinary cell from the body and transform it into a cell that's very similar to an embryonic stem cell. It's called an induced pluripotent stem cell, or iPS cell, and it can be made into all the cells and tissues of the body. All that's required is to add a few genes that reprogram the cell back to an embryonic-like state. These cells could possibly be used instead of cells derived from human embryos. But bringing this method into medical use is another story. First of all, the current technique for making these iPS cells is very inefficient. Less than 1% of cells actually fully reprogram. A more important concern has been safety, particularly a risk for cancer.
Standard protocols for making iPS cells involve the use of viral vectors which put DNA into the cells to affect reprogramming. This is potentially very bad because this can sometimes lead to activation of cancer-causing genes.
But most important of all, it's extremely difficult to create different kinds of tissues from iPS cells that could be used for treatment.
In order for this technology to be translated to patients, we have to envision generating clinically useful cell types.
Derrick Rossi of the program in Cellular and Molecular Medicine and Immune Disease Institute at Children's Hospital Boston led the development of a new technique that could solve all of these problems. In the current technique, a virus carrying the 4 key reprogramming genes is allowed to infect the cell. The reprogramming genes become a permanent part of the cell's DNA, its genetic material. A messenger RNA is made from the gene and travels to the cell's ribosomes, and the ribosomes use the RNA as a blueprint for making the reprogramming proteins. The cell is transformed into a stem cell. It still carries the introduced genes and will pass them on to its daughter cells every time it divides. The new technique is much more direct. The researchers create the messenger RNAs themselves, but specially modify them to prevent the cell from mistaking them for a virus and attacking them. These modified RNAs are introduced directly into the cell and go straight to the ribosome, where the proteins are made. The cell is transformed into a stem cell, but its DNA is unchanged.
By using an RNA vector, we completely eliminate the risk of causing cancer because RNA does not integrate into the genome of the host cell. By being non-integrative, you've got a huge safety advantage, yet you get the same benefit of being able to add proteins that are missing back to the cell.
What's more, the stem cells made from modified RNAs seem to resemble embryonic stem cells even more closely than those created with genes and viruses.
Our data suggests iPS cells made by the RNA approach more closely recapitulate the molecular properties of human embryonic stem cells than iPS cells generated by viral methods.
The Rossi Lab's new technique also cut in half the time required to create iPS cells. The researchers are most excited about what they can do with these cells once they're made. Using modified RNAs, the newly created stem cells can quickly be transformed into the different kinds of cells a patient might need.
So the pluripotent stem cell is just a start. It has the potential to give rise to all the cell types, but it has to be directed towards that fate. That has been typically done by modulating the environment around the pluripotent cell. What our technique does is eliminate that whole approach by adding in factors directly into the cells which drive its fate very specifically down to different clinically useful cell types.
Dr. Rossi is cautiously optimistic that this technique could be generally useful even beyond the stem cell field, allowing scientists to replace any protein that's missing in patient cells.
In terms of therapeutics, any genetic disease that involves a mutation of a gene that doesn't make a certain protein, we can now approach that with this technology to reintroduce that protein into those cells and reestablish proper function to those cells. So we think that this is going to be really important for many avenues, therapeutic avenues, in addition to basic questions of biology.